Well, I'm pleased to introduce our first guest on my new podcast, based with Antic, Professor Nikolai Petrovsky, a man qualified to discuss viruses and vaccines if there ever was one. Professor Petrovsky graduated from the University of Tasmania with a Bachelor of Medical Science in 1979 and a double degree in Medicine and Surgery in 1982. He completed a PhD in immunology, focusing on cytokine regulation in health and disease, and from 1998 to 2004, Professor Petrovsky was the director of the National Health Sciences Centre in Canberra, the clinical research organisation, by the way, where he conducted clinical trials, and he's been the director in the Department of Diabetes and Endocrinology in Flinders Medical Centre in South Australia ever since. Now, in 2002, he founded an SA-based biotech company specialising in vaccine development and trials, and since then, he's played a key role in the development of many vaccines and has contributed to over 220 peer-reviewed articles to various academic journals, as well as holding editorial positions for various other scientific journals such as Open Immunology Journal, the Journal of Vaccines and Vaccination, and more recently, Professor Petrovsky developed a vaccine which has been authorised for emergency use in Iran for COVID-19. There's much more that could be said, but welcome to the podcast. You have the esteemed honour of being the first guest on our podcast, and also the crash test dummy for it. So welcome, and I brace in for the inevitable ride. But I want to ask you a heap of questions now. It won't surprise you that there's a fair bit to cover because, of course, there's a lot of history in all of this. But I want to start off. Tell me about your vaccine that you created. Where's it up to? What's the status? What's the lie of the land?
Okay, so our vaccine is now an approved product in the Middle East. Mm-hmm. We've delivered with our partner Synergen 8 million doses, so which is no mean feat.
That is no mean feat.
And the great thing is that we've seen very positive effectiveness, particularly against preventing severe disease, which is what we're trying to achieve with these vaccines. We shouldn't be pretending they stop infection or transmission. They never did. They never will, probably, at least the current generation. But what they are very good at is stopping people who are at high risk from getting severe disease. Yeah. So that's what we showed with our vaccine. And along with that, we had great safety data. Yeah. And you can't overemphasize that. What kills most vaccines is not whether they work, but are they sufficiently safe to give to millions, if not billions of people? So safety is everything in the vaccine world. And so, we're obviously very reassured that having handed out millions of doses, we haven't seen any of the problems that have been at least theoretically associated with some of the other vaccines, particularly the heart inflammation, the myocarditis, the severe blood clots and what we call central vein thrombosis, which can lead to death, and then some of the autoimmune problems like Guillain-Barré syndrome. So again, we never rest on our laurels. The job is never done. What we can say is that, we've given it to millions of people and we haven't seen any of those signals.
And is there, the process was dealt with by the regulatory body of the country that it's using it now?
Yeah, so the way it works is that each country has their own regulator. Yep. They all work in essentially the same coordinated way. Yep. They ask the same questions. And typically you go to the regulator of the country in which you've done your major, what we call pivotal phase 3 study, which is, in our case, was just over 16,000 people were in the study. And so because the regulator in that country is intimately involved in that study, so they actually can see the data as it's being generated. So if there's nothing being hidden, and so they obviously have a lot of confidence that it's not just a dossier being given to them, but they've seen inside the study, they've seen what's been happening, they've talked to the investigators, and therefore they have the confidence that, yes, this is all working as it should. Yep. And so not surprisingly, you get that approval there first.
And this is a very vigorous process, a very rigorous and vigorous process and a similar one to other countries, as you say, in this country. I mean, it's a carbon copy, if you like. So if it's passed this test, it's passing any tests. or approval process?
No, exactly. So because people always ask, well, maybe regulators in some countries operate to different standards.
Right.
And, if you go to, say, a developing world country, I mean, do their regulators operate at a lower level?
Right.
It actually is quite the opposite. It's in reality, what you find is that the smaller the country, the more nervous the regulator is, the harder a time they give you, the more questions they ask and the longer they take.
Yeah, right.
And so ironically, often Australia will be, and the US, Europe, will be the first countries to approve a new product because in a way they're more confident. It's not that they hold things to a higher standard. In fact, you could argue what we've seen, at least with COVID, is they've done the opposite, that maybe they've held things to a lower standard than what people are expecting. But it isn't true to say that, you get an approval outside of those three countries, it's somehow at a lower level.
Yeah, it's almost counterintuitive in a sense, isn't it? And I want to get back to the approval process and some of the other stuff you mentioned on there before, but just about in terms of your, and don't feel shy about telling us, but I want people to understand the significant expertise and background, 220 peer-reviewed studies that you've authored or been co-authored of, but just a little snapshot of the history and your qualifications to get to this point. Like you've got a world-class background in my view.
Well, thank you for that. It's been a long journey. So I did my PhD some 2 to 3 decades ago.
You wouldn't know.
It's too long. It's in the last century. Yeah. So it was before a lot of people who I guess are in the audience were born. And after that, I was a practicing clinician, and I remain a practicing clinician, which is why, to me, safety is everything. Because we want to look after people and we don't want to do harm. But I was passionate and fell into developing vaccines after my PhD 25 or so years ago. And fortunately, I was successful in obtaining funding from the US government, which is unusual because we are not a US-based enterprise. All my team is here in Australia. So we're one of the few groups in the world that the National Institutes of Health in the US fund outside of the US. Now, we would hope to think that means we must be doing something really special. And maybe the first time they funded us, people would say, well, that was a fluke. It was after 9/11 and, George Bush was trying to really move vaccine science forward. But, we have been continuously funded by them for the subsequent 20 years in larger and larger amounts. And I think in total now it's over $50 million that they've put into our program. So
the US government.
The US government. Not the Australian government. The US government. We're an Australian outfit. A little bit unusual, but again, it is what it is. It is
what it is.
But, as a consequence, we've been fortunate to be able to use that funding from the US to develop vaccines against the SARS coronavirus in 2003. In fact, that was one of our first programs. Very successful, protected animals against a lethal infection. We then had the opportunity to apply it in 2009. You may recall we had a thing called the swine flu pandemic.
I recall it.
It wasn't as severe, and it spread and everyone got it, but it wasn't severe. And again, people confuse pandemic or the label of pandemic with severity and lethality. And in fact, there is no connection. So a pandemic is a new virus that is going to infect very large numbers of people in multiple countries. So it's more about a new virus spreading through global populations than it is a virus that's 80% lethal or 1% lethal, or in the case of COVID-19 currently, well under 0.1% lethal. The lethality doesn't come into the definition. And so, when we worked with SARS and then we worked with the swine flu in 2009, we actually had the first vaccine in the world, in humans, 3 months after start of the swine flu pandemic. So we were first in the world.
Well, you think that
again, yeah, you'd think
something to celebrate.
That would be something to celebrate, and that would give you kudos.
Yeah.
And that was the fastest human vaccine development in human history.
Yeah.
Like it was like around 3 months, between discovery of the virus and when we were starting our trials at Flinders. Very successful trials. And subsequently we did the MERS coronavirus in camels, successful vaccine, again, another coronavirus. So when it came to COVID-19, we'd done this so many times before. To us, we said, look, this is self-obvious. We need to be diving in here and making a good vaccine based on all these good vaccines that we'd developed in the past. So to us, it wasn't something totally new. We'd had all this background experience. So we thought we sort of have a duty to produce a vaccine and produce a really safe and effective vaccine, because we probably know more about this than anyone in the world, without sort of trying to sound egotistical, because we've been doing it for 20 years. We've been doing it in humans for 20 years. We know what we're doing. We have the tools. All we need is the assistance. And we can get this out there and giving it to people. So that was the motive to push on.
And your vaccine's quite different to some of the others. I just for a bit of background, can you just break down the main types? So the main types of vaccines in their technology brackets.
Okay, so let's do it by sort of age of the technology. So the very original vaccines, over 100 years old, are what we call the inactivated viral vaccines. And so in this case, you take the virus, SARS-CoV-2, and you infect cells with it. They make lots more virus. You then purify that virus. You kill it with something like formalin or a chemical that will kill the virus, and that's now your vaccine. And in fact, most of the vaccines in China and in India are actually that 100-year-old technology.
And that compound that's left, or that material that's left, then tricks the immune system into producing the reaction, the immune reaction.
Yeah, because you're giving the virus. It can't sort of make you sick because it's been killed. Yeah. But it has all of the, hopefully, features that the immune system can recognise and make antibodies to that will neutralise the real virus. Yep. Now, it's, as I say, it's a very old technology. It does have a few problems. One is that because you're working with the real virus, you have to have very high level biosecurity around your manufacturing facility. Yep. Now, there is no manufacturing facility really in Australia that could do that at large scale. And so it was very limited. I mean, there were facilities in China and India, obviously, but not many other places. But so that's the sort of traditional, our flu vaccines, the seasonal flu vaccine that a lot of people get. That's how it's made by CSL in Melbourne. Yep. They grow up the virus, in their case, in eggs, kill the virus, and you just inject people with that killed virus.
So the dead virus vaccine, that's one.
Yeah, so that's one. Then the next one, in terms of I guess, age, is in fact the one approach we're using, which is called recombinant protein. Right. So in this case, what we're doing is we identify from the virus, which the virus is made up of lots of different proteins and other components. But generally, there's just one protein, which is the protein the virus uses to attach to your cells, which is key to the ability of the virus to cause the infection. And so if we can just identify that one protein, we now actually synthetically make that protein. So we don't deal with the virus at all. We're just making a single protein from the virus, and then we're now injecting you with that protein. So your body will recognize the protein and make the antibodies that now can actually recognize and block the virus. So that's, for instance, the hepatitis B vaccines, the human papillomavirus vaccines. A lot of the childhood vaccines are based on that synthetic protein technology.
And these are done with insects.
So the difference
you're gonna eat the bugs.
Yeah, so we only need one cell from an insect. So these are immortalized cells. So we don't work with the moths themselves. But so this cell was taken from a moth 30 or 40 years ago, and it's been growing ever since in a broth. So it's an immortal cell line. And so if you look at it, we just have a big vat of broth, and in it are floating just these single cells.
Yeah, right.
And so we then infect those cells and get the gene of the spike protein into those cells, and those cells are now tricked into producing and secreting lots of that protein, which we then purify. So the beauty of this technology is it's unbelievably safe, because you're just working with a protein. You're not working with a virus or anything else. It's highly pure, so it doesn't have a lot of side effects in terms of, making people get fevers, which you can get with the inactivated viral vaccines. They tend to be a bit dirty, as we call them. So it's not unusual to get some people having quite a high fever, for instance, after it, whereas we don't see that. So it's a very pure, defined, safe and effective process. And so we thought, and have always thought, that is the best approach. The challenge is not many people can do it. It's really tough to make a synthetic protein, because proteins are big and they're complex.
Right.
Even though you're tricking these insect cells to make them, you still have to purify the protein. You still have to make sure the protein is all folded in the right way, which is not easy.
Is that a technical thing, or is it an infrastructure thing, or is just complex?
It's a knowledge thing. So it's technical and knowledge. And if you have all the conditions right and you know what you're doing, which obviously we believe we do because our vaccine has been highly effective. Now, Sanofi, which is the biggest vaccine company in the world, was given $7 billion by the US government to do exactly what we've been doing, and they didn't get it to work.
Right. Yeah.
So their vaccine hadn't been approved, right? They'd had to go back to the drawing board and try and fix some of the problems, despite having unlimited resources.
Yep.
And here we are with a tiny team completely, a year in front of them, because we knew what we were doing and we got it right.
I was going to ask you that question. So you put that down to that's just knowledge. That's IP.
It's the people, and those companies have tens, if not hundreds of thousands of staff, but we have like 15 staff. But in some ways, as long as those 15 people really know what they're doing, or even a few of those people really know what they're doing, they can achieve more than 10,000 people who are all in a big organisation, but no one really knows what they're doing.
Well, you sound like you're talking about a bureaucracy there to me. We would never say that a bureaucracy would be inefficient, of course, so because there's so many good ones. I can't name any of them. Anyway, putting that aside, so also from there we've got now vector. Have I jumped the gun?
Yeah, no. So the next category, which has been around for a number of years now, is what we call the viral vectors.
Viral vectors, yep.
And so what we're dealing with here is live viruses, which always, as a clinician, always makes me a little bit uneasy, because whenever you're dealing with live things, there's unpredictability.
Right.
You have a dog. It's a live thing. You might say, my dog's friendly and it's tame and it will never do something wrong, and then it bites someone. Inherently live things are unpredictable. And so live viruses, even, when they're a genetically modified vector, there's always an element of uncertainty, and it might be small, but you can never extinguish it. And so most clinicians are, I think, to a whole, are prepared to accept, yes, this has been characterised, but they're always just that little bit nervous.
A bit nervous about it.
But so what we have here with the viral vectors, and I think, again, the important thing here is we had completely new vectors that were being used. So if you've had a viral vector and you've been using it for 30 or 40 years, you've grown to understand its behaviours, and that builds confidence. I think, again, where a lot of nervousness came with what was happening in COVID is completely new vectors were being used for the first time. And so the uncertainty around that is much, much larger.
Yeah.
And so, we suddenly saw after the vectors were rolled out, so these are the adenoviral vectors like AstraZeneca, we saw this side effect of, thrombosis and idiopathic thrombocytopenic purpura, which is loss of platelets in association with blood clotting. Completely unexpected, that was linked to the vector, because we now know it's occurring with the different adenoviral vectors out there. It's not occurring so much with any of the, like the inactivated or the recombinant protein. It is occurring a bit with the mRNA, which is interesting. There is an increased risk of thrombosis that's sort of not been as prominent as myocarditis, but it's there. But again, the problem is you use something for the first time with not a lot of knowledge about it, it can come back to bite you. And I think that's what we saw with the vectors.
Yeah, who would have thought? And mRNA
Well,
vaccines?
finally. The newest and technology none of us was expecting to see. We'd all worked with it.
Yeah. So tell us a bit about that for start off with, because you know a bit about this area. You've worked with mRNA vaccine technology, so as well placed as anyone to tell us all about it. What's the history first up?
All right. Well, so mRNA was really introduced around about 15 years ago with an idea of gene therapy. So an idea of treating people who have genetic diseases. If you can inject them with mRNA, then that mRNA will program their cells to make the missing protein that's, say, causing the genetic disease, because most genetic diseases are lack of a particular protein because the gene's not working. So mRNA was never introduced or thought of that it was going to be a vaccine technology. It was a gene therapy technology. And so it was rolled out by a number of companies, and they went into human trials with it as a gene therapy, where unfortunately it had failings. And one of them being that if you're going to give a gene therapy, you have to produce or replace that protein indefinitely. And the mRNA gets the cells to express the protein, for weeks. Now we know at least months, but almost certainly not for 10 or 20 years. So you're going to have multiple doses. It was only, in the last 5 years or so that company, and particularly Moderna, sort of repurposed and said, we think we can maybe make a vaccine with it. And at that point, we said, okay, we better look at this technology, and assess it ourselves, because maybe we need to
be on the cutting edge,
be at the cutting edge and move over. So we developed our own mRNA vaccines. We tested them. Ultimately, we decided no, it's too early. It's not effective enough. There's too many unknowns, particularly on the safety side. It's going to take 10 or 15 years to collect the safety data. I'm hoping to be retired in that time. For a pandemic, you want something reliable that's certain, and recombinant proteins the only way to achieve that. So we actually put it to the side, said maybe in 10 or 15 years it will be ready for prime time. Maybe not. Maybe it won't work, but it isn't ready now. So, and many of us in the field came to the same conclusion. So we were the most stunned of all when, 12 months later and they're saying, give it to everyone, give it to the whole world's population. We're going, hey guys, I'm getting really nervous here.
Hold fire. So just to explain what that actually does, because people, it's technical stuff, but in essence, that is it's an injection filled with this mRNA, which is a messenger RNA compound, or I'm using the wrong language, which basically tells cells to produce a spike protein, to produce the what's the word I'm looking for? The compound that looks like the virus.
So I think just to explain genetics 101
properly.
So our cells, or the way our genetics is based, we have DNA, which is an alphabet that encodes all of our genes. And that DNA inside our cells has to be turned into RNA, which is like a reading off one alphabet and translating it into a second alphabet. And then the RNA tells ribosomes, which are these protein complexes, to make proteins.
Right.
And that makes our cells, it makes us what we are.
Yep.
So mRNA is again an alphabet or a string of letters in a code that are all strung together. It's formed by sugars, but in essence it translates DNA into a set of instructions that tells our cells what proteins to make.
Right.
So if we take any mRNA, even a foreign mRNA, and get it inside the cells can't distinguish the foreign mRNA from the mRNA that's in there all the time that's giving it instruction. So we're tricking the cell now to make any protein. When it was gene therapy, it was trying to get them to make a protein that they should be making anyway. In the case of a vaccine, essentially you're tricking the cell to make the spike protein that would otherwise be made by the virus.
And then that then causes the immune reaction. The body thinks it's had it before, and you have the same effect. You have a dumbing down of the effect of the virus when you catch it.
So what essentially we do when we make a protein, we inject it, stays outside your cells. The immune system, the cells of the immune system, particularly the phagocytes, eat that protein.
Right.
And then they present it to what we call T and B cells, which are the cells that say, oh, this protein is foreign, must be associated with a virus or a pathogen of some sort. So we better make an immune memory, like make antibodies and T cells that recognize it.
Right.
When you do it with mRNA, so the mRNA itself isn't recognized per se, it's just an alphabet. So it's a program. And so that program gets inside the cells. Those cells make the spike protein, which now gets into the surface of the cells, into the membrane, and gets secreted from the cells. And at that point, it gets recognized by the immune system. But it's similar, but it's not equivalent to injecting the protein from the outside. Because now the protein is inside your cells.
Yeah.
Which is very, very different.
Very different, and very complicated as well. There's obviously a whole range of things that come to that, but I'm just interested in the actual virus itself. Obviously, a lot have been made of the origins, but I think I'm right in saying that you developed a view pretty early on about the origin of the virus, if I'm right in saying that.
Yeah, so one of the things we've been doing for 20 years is we were the first real adopters of artificial intelligence in this field. And again, that's one of the reasons we've been successful in getting support from the US, is we're always trying to stay at the cutting edge. So when this new description of a new virus occurred in China, we knew nothing about it. They knew nothing about it. No one had even got an isolate of the virus, but they managed to sequence it and get its alphabet. So you just got a big string of alphabet. We actually used our artificial intelligence to translate all of that to work out what actually was in the virus. And the idea then was, and we designed a vaccine based on an alphabet. No one had even characterized the virus at that point. But alongside that, we thought, well, is there anything else we can do with our artificial intelligence that might be of interest and of use? And because at the time there was a lot of debate going on, where did this virus come from? and everyone assumed it came from an animal source, as did we.
The wrongly framed pangolin.
Yes, the pangolin.
Pangolin did
we'll
nothing
get
wrong.
to that. So we thought, well, this is great. We have a model of the virus in our computers,
Yep.
and that we can model all of these different species of animals, and then we can actually determine in our supercomputers, well, what species of animal matches best with the virus? Because that would give us a good idea of where this virus came from. So we looked at a whole range of different animal species that might have come from, including the pangolins. They're there, but we also looked at civet cats, which were the source of SARS-CoV-1.
Yes.
We looked at bats. And then we did the usual, cats, dogs, cows, and basically then analysed, well, what was the best match for the virus? Right at the start of the virus. Humans came out on top. That was not what we were expecting. And so you're scratching your head, so this is a perfect human virus that didn't come from humans. That presents a fundamental conceptual problem. How did it know how to become a perfect human pathogen if it had never seen a human? And so you start to think, well, is there any evidence it was circulating in humans, just not recognised? And certainly, even within that first few weeks, there was no evidence this virus had been circulating in China or anywhere else, unrecognised, to adapt to humans. So you're thinking then, well, how else could it adapt to a human if it wasn't in a human? And these are just sort of, thought experiments, so no presumptions. And we're going, well, what about if it had been in a culture with a human cell or with a monkey cell, it could adapt in the culture, just like we do with our insect cells. People, can have cell cultures of human cells. We all use them in our labs. What about if this virus, somehow got into a culture well and they don't know it's there, but they're culturing the human cells, the virus is being carried along and this happens, and this is, again, not something unique. A lot of cell cultures, 50 years after people have been working on them, they suddenly identify a virus as being there for the last 50 years, propagating in those cells. They just weren't aware of its existence. It's silent, So we thought, well, no, that's a perfect explanation, because it doesn't even require a human to know it's happening. It could be just, some cells from a pangolin which were infected by the virus were cultured in the same lab as had some human cultures. The virus managed to cross over just because of cross-contamination. So we're suddenly going, okay, so a lab, this could have come from a lab. Not saying anyone has deliberately made this virus, but it's completely logically consistent with everything we knew about this virus. It was perfectly human adapted day one. You know what I mean? It has these features that suggested it's been adapted on at least human cells, if not humans. Are there any labs where it could have come from? Well, turns out Wuhan has the major labs in the world that work with coronaviruses and coronavirus samples, from pangolins, from bats that have human cell lines. You're going, okay, well, that's pretty plausible. Again, not suggesting any knowing human involvement, but just looking logically. Then you say, well, where did the pandemic start? Was it in the vicinity of any lab that this might have come from? And then you realise that, the Wuhan Institute of Virology, for instance, is only a couple of kilometres from what appeared to be the very epicentre of the outbreak. And on the same train line, it's one train stop from the epicentre. And you go, so, someone got infected in the lab, again, accidentally, went home on the train line to the next stop, got off, went to buy some groceries at the wet market to make dinner, transmitted the infection to a store worker there, who then transmitted it to people. And you go, okay, this is all plausible. It's not a conspiracy theory. As a scientist, all you're trying to do is break things down and say, is this possible or is it not? Is there any evidence to support or not? So we entered into this completely innocently, saying, well, we've got this great modelling data, which to this day no one has refuted on, and in fact all of the subsequent experiments that were done in China and other places established that all our modelling was 100% correct. Yeah. That, there was no species that showed a better fit to this virus than humans. So again, none of that's in dispute. We, again, were open-minded about how to explain that observation. So we said, let's publish this. This should be really high profile. Everyone's going to want to see this. We were just knocked back time and time. It was being censored. This is a scientific paper, not making any assertions, no conspiracies. We're going, what's going on? We've now submitted it to, Nature, all of these leading journals, and it's sometimes bouncing in 24 hours. Basically, we have no interest in this. And you're going, there's something going on at a level we don't understand. This is not a coincidence. It was very rigorous, the paper. We had experts look at it and critique it. And as I say, and all the experimental data fitted it. And yet we couldn't get it published. It took us over a year to get, and this was really important data early in the pandemic. They managed to sabotage it, so by the time it was published, it was almost old hat.
Yeah, right.
Do you know what I mean? But it was obviously quite deliberate, and at that point we started to realise we're up against something we'd never seen before.
I mean, there's so many questions that arise out of that. I just want to, and I don't know if you can feel comfortable answering this, but how do we know that same sort of freezing effect is not happening on adverse effects, and other sort of studies that are being done, have been done, might be done through the similar journal line at the moment? I mean,
well, we know. It's not a question. I know multiple authors now around the world because we obviously have ended up connecting to people and sharing stories.
We're talking about,
and this is systematic censoring. And I know obviously it's happened in social media. I think Elon Musk and Twitter have exposed. The level at which this has been operating. But also, just like it's been happening in social media, it's been happening at the very core of the scientific establishment.
You see, this is the thing I think that people need to understand is just how much of this has been buried over the course of the pandemic, but beyond. The criticism I always get, I'm jumping off track here, but I don't want to do that, but the criticism I always get was, well, show me the peer, everyone knows the term peer-reviewed now. Every layperson like me now knows the term peer-reviewed, so we all use it. Show me the peer-reviewed data and the reports and all that sort of stuff. We know now from what you're saying and from other evidence that we're saying that this is actually getting physically buried, beginning with what you're describing, but also studies on adverse effects of the current round of vaccines. I mean, that's a real thing.
It's a very real thing. And when you say buried, so, our research was finally published under peer review. There was nothing wrong with it. But they managed to hold it up for over a year. So during that time, no one has access to that information. Right. So when you say, they're saying, well, show us the peer review data, all they have to do is hold it up for a couple of years, and by then everyone's forgotten and moved on. Yeah, right. So it was a very deliberate campaign, not necessarily to stop it ever being published, but it was to stop anything that they didn't want being published during that time, for the first year or two, during which they were trying to push a singular agenda and they were trying to censor anything that questioned that agenda and that narrative. And I think that's applied to across medicine, science, social media.
I was going to say that.
It was totally coordinated. And no surprises. And we even know where it comes from because, there were workshops that decided to do this that were held before the pandemic started. And this is just the game plan that they had already decided they were going to enact, if and when there was a pandemic. So it's not a conspiracy theory. This was a strategy that's public. that's been laid out, that's there for everyone to see. How it's been applied has been horrific. Because I think at the time they were thinking, we're going to have a global pandemic, billions of people are going to be dying. They were thinking a scenario that is a million miles away from what we've been dealing with for the last 3 years. Unfortunately, they when they were planning this, they didn't say, but we have to modify this if billions of people are not dying, because what we're planning to do is take away everyone's rights globally, right? We're basically going to have a totalitarian system where individuals have no rights, doctors have no rights, scientists have no rights, because we're trying to save humanity. Well, humanity wasn't at risk, let I say it, in the last 3 years. Yes, people have died with COVID. Humanity has never been, the only thing we've been at risk of is what's happened by this totalitarianism, because that is scary and that can have long-term consequences that may be much more horrific for humanity than SARS-CoV-2. SARS-CoV-2 is a problem, but it's not going to eradicate humanity.
That's a really
It's about
good point.
getting it into balance. This was never in balance. The things they had decided they were going to do were so out of balance with the problem we were dealing with that I think the consequences of their actions have been catastrophic. The consequences of the virus actually are probably quite benign by comparison.
Well, let's talk about that. So the virus itself, so we've got multiple strains that have come through, and I've used my lay eyes to look at one of the, there are sites that track the various, and it all gets a bit too complicated for me as at the point of having to type in the website address, but it's a very complicated chain, family tree of variants, and so on and so forth, and they go on. Some become variants of concern, some don't, but we start with the original strain.
Yes.
Let's talk about that. How dangerous and virulent was that? And actually, there was one question I wanted to ask you on the way through there, which is, in your mind, a person that's a single person that's exposed to a virus in a lab, does that person walking out into the community unrestricted have a real prospect of spreading the virus in the manner it is purported to have happened? So, for example, if a lab worker was exposed to the virus, went out and did his shopping as you previously described, is that a plausible scenario for a global pandemic? Is it enough to leak in that manner?
No. So what's really interesting, and bird flu is the classic example. So here is avian influenza, highly lethal. If you get infected by a sick bird, then your chances of dying from that infection are around 50% to 60%. But it hasn't caused a pandemic. So what happens is each time a sick bird person comes in contact, they get infected. They have a high risk of dying. They get very sick, but they don't transmit. And so that's because it's still a bird virus. It's not a human virus. That's what's different about COVID. It was a perfect human virus before the first infection in humans. So it's not a normal pandemic virus. Anyone who claims it is not telling the truth. Most pandemic viruses, so they're trying to cross over between species. And they try and they try and they try. And bird flu, it's been doing it for 20 years now. So we've had several thousand people die, but never has it found the trick of being able to now cross from one person to another. It hasn't, because that requires human adaptation, and if it's only caused a few thousand human infections, there haven't been enough opportunities for it to find the trick of spreading from human to human. So if a lab worker is working with a bat, and again, that was what was happening in Wuhan, and some people said, that must be how it happened. It was a bat infected a human. Well, bats infect humans, in rural villages, we know, in China and other places, with lethal viruses, and some people in the village may die. But again, those bat viruses can't transmit human to human. So the plausibility that this was a lab worker getting infected by a virus from another species that they were culturing doesn't cut it. The probabilities are infinitesimally low. The only way you can plausibly say this could happen is if they took the bat virus, put it into human cells, culture it with the human cells, and adapted, basically taught it how to infect human cells, which now gives it the ability to spread human to human. That is what has to have happened, and what, you know, many people now believe did happen. Whether it happened deliberately or whether it happened accidentally, it still had to have happened that way. There is no plausible explanation for how this came from a wild animal, got into the middle of Wuhan, you know, with no one seeing anything, adapted to humans, and then started a global pandemic.
But is it plausible for one, taking aside where it started from, a person that's infected with it in the lab, from wherever it came from, then walking out into the community unawares? Is that a plausible scenario for a global pandemic to start? Is there enough spread from a single person walking around
Oh, absolutely. No, so if someone's walking around with a transmissible human-to-human virus,
it could shoot off.
And we know, I mean, we actually know the statistics. So with Wuhan, an infected person infected 2.5. It's what we call the R0. So what the R0 tells you is how many people on average does an infected person infect. So the R0 of Wuhan was calculated from the number of people infected in China over time. So it was around 2.5. So one infected person, so one infected lab person who got infected by their culture, right, on average would have transmitted it to 2.5 people. So those, you know, but it could be more, it could be 10, or it could be 1. It averaged 2.5. Now, you think, oh, that can't start a global pandemic and infect billions. But we're talking exponentials here. So if you put, you know, 2.5 to the power 10, you know, what number do you get? If you put 2.5 to the power 64, you know, you have a number that is greater than the number of grains of sand that exist on this planet. You know what I mean?
That's a timing issue.
That's just 64 transmissions.
Right. Yeah. Oh, no, certainly.
So it's like 64 doublings. Now, you know, that happens. Say you infect someone, and we know with the Wuhan strain, it was about a week before people became infectious. So the doubling time is a week. So the first week it's one person, the second week it's 2, the next week it's 4, the next week it's 8. Sounds low. Go a few more doublings and you're now 1,000.
So do those numbers marry up? So from the timeline that we expect from when we think it came from walked out of the lab or whatever to where it ended up in February of 2020. They all marry up. That's all. Yeah.
So if you work backwards from what we believe to be the true numbers in China, and unfortunately the numbers from China have never been believable. Right. But from other estimates that we can make of what was happening in sort of January 2020, you can sort of do your halvings and work when was the first case. And when you do that, and you can also do it genetically by looking at genetic variations, but whenever you do that, you map it back to September 2019. So sometime around September 2019, the very first person walked out infected, and from there it all fits beautifully with the mathematics.
And so that first strain, in terms of how dangerous was it, the very first, the one that walked out?
So again, you know, we saw a lot of misleading data. And so what you have to remember is at that very early time point, when you're talking about number of people infected, from which becomes the denominator, well, how many people were infected, how many died? You do that ratio, it gives you what we call the case fatality rate. So, but the problem was that, you know, they were only looking at the people who were in hospital, so the sick people, and assuming they were all the cases. And of those cases, say 5% were dying. So they say, oh, the case fatality rate, you know, it's pretty high, 5%. You know, that's scary. Not quite in SARS or MERS or, you know, pandemic influenza sort of scary. but pretty significant if 1 in 20 people are dying. The problem is that wasn't a true number because, they're only looking at the very sick people and using them as the denominator, not everyone who was getting infected. And that's why the Chinese numbers are so important, and the fact that they're completely unreliable is so important, because that's how we were getting our denominator.
Yeah.
When we now look back and say, okay, so the Chinese were claiming, there was 100 cases, and in reality there was maybe, 5,000 cases. Including people who didn't have symptoms or people who were not going to see their doctor. And now we divide the same number of, 50 deaths by now, 5,000, and we go, oh, actually the case fatality rate's 0.1%. Not much different to seasonal flu.
Well, that's the question,
And so those early big numbers on case fatality rates were completely misleading and allowed this preordained campaign of censorship, right, and totalitarian sort of control over the narrative to be rolled out, because it was rolled out on the assumption this is a highly lethal virus. But in truth, it was, and again, it was a whole series of comedy of errors. The fact that Chinese were not giving true numbers meant people were calculating the wrong case fatality rate. Because they were calculating the wrong case fatality rate, they made the wrong decisions. And so all of this happened.
Who would have thought the Chinese Communist Party would get their numbers wrong? That's extraordinary to me, that statement. Just on that subject then, so if the progressive incarnations and variations, mutations, then have become even less virulent as they've gone. Obviously, Omicron was a mild strain, but even the Delta and I think the Beta strain as well.
So in fact, arguably, so Beta and Delta and Gamma and again there's a
family tree.
There's a whole lot in the middle there.
It's like the Addams family.
They were much more transmissible than Wuhan.
Right.
So progressively, with each step, their transmissibility basically doubled. So the R0 is going from like 2.5 from Wuhan to Beta, around 4, Delta 5, 6. Omicron is just blown. It's now the most transmissible human virus that exists, even greater than measles. We've never seen anything like this before. So they're more transmissible. In those early ones, no, they actually, if anything, the lethality maybe went up. But we were starting to get truer numbers of our denominators. So the reason the case fatality rate looked to be reducing was not because the virus itself was more benign going to Beta and Delta. It probably was actually more virulent. But we were getting better senses of what the true denominator was because we were starting to get tests. So we got the PCR test. We could go and look for people who were infected but weren't symptomatic. And so suddenly we're getting a better denominator. And so ironically, I think, that early case fatality rate coming down was again just a false nature of the numbers. The virus was getting more lethal, and probably Delta was the most lethal of all of the variants we've seen. But then we had a big step down to Omicron, where clearly it was a lot less. It was more transmissible, but less lethal than Delta. And there were probably a number of factors, not just the virus at that point in time, that were contributing to that lower lethality. Because by the time Omicron came on the scene, in a lot of countries, in fact, at least 50% of the population had already been infected by these earlier variants. So, of course, they're not, even if they get Omicron, which they still could, they weren't going to get very sick. So that might artificially make Omicron look more benign. But if you'd never been infected and got Omicron and you were over 80, the mortality rate is not that different to Delta. And so, the idea Omicron is completely benign is again a bit misleading because it's still killing people.
Of course.
But it's killing the people who are the most vulnerable. if you or I, well, I'm sure we've probably everyone in this room and everyone in Australia has now had Omicron. I mean, that's what the statistics show if we test. And we're all here. We're all well. You know, maybe we had a day off work, maybe we didn't have a day off work. You know what I mean? It is a pretty benign infection, fortunately.
And so, I mean, obviously it does have that effect on the elderly. We'll get to the issue of, the risk-benefit analysis of making younger people take an experimental therapy. We'll perhaps come to that in a minute. But just in terms of that, and I know this is a really complicated area, it's probably hard to distill down, but is that what you'd expect for a mutation sequence for a virus like that? Is there anything unusual about the way in which this has mutated this virus? Or is that as expected? And do they burn themselves out, these viruses?
So no, they don't burn themselves out, unfortunately. No. They do adapt. So yes, this was all completely predictable. And we were predicting this, you know, in the first weeks of the pandemic, that, you know, this will mutate. It's an RNA virus. RNA viruses always mutate. Flu is an RNA virus. Every year you have to have a different vaccine. We're already predicting vaccine failure before we even had a vaccine. No one was interested in listening, unfortunately, but the science, again, wasn't rocket science. Because we understand the nature of these viruses, and although SARS-CoV-2 was a new variant of these types of viruses, it still behaves like other coronaviruses, other mRNA viruses. So the fact it's formed all of these families, completely predictable and expected. The fact that it may attenuate, which become less lethal, is interesting, because it some people would imply that will always happen, but in fact it doesn't. So the way viruses evolve is that they are looking to become as established as they can in a new population. If they kill too many people, that takes away potential hosts. It's not good. That, you know, they may burn themselves out if they kill most of humanity. Who's there left to infect? So a virus like Ebola, you know, that has 80% lethality may well, you know, almost certainly will attenuate if it has enough opportunity to get. Then it might reach a perfect sweet spot of 5% mortality. Then there's no benefit going lower than that, because losing 5% of your hosts, so what? You've got 95% left. So the idea a virus will attenuate itself away out of existence is a nonsense. It will, if it's too lethal, it will attenuate itself to a point where it's not killing too many hosts, and so it still has feeding ground, and that's where it will generally stop. Like influenza. So influenza, you know, the current H1N1 came from the Spanish influenza, same virus, right? This is its progeny 100 years later. So Spanish influenza probably killed 20% of people it infected. You know, the H1N1 that causes seasonal flu, you know, it's around 0.1%. So it's found a sweet spot. And over the last 100 years, it goes up. Some years it will cause 0.3% mortality in a bad year where it's found a different mutation. Some years it will cause less. But it's around its sweet spot. And so COVID, we predicted, is going to follow the same trend. It will find a sweet spot where the lethality is not going to deprive it of a host. And at that point, it will probably vary variant to variant and season to season. It could go up or it could go down, but it's not probably going to go to 100% lethality, and it's certainly not going to go to 0% lethality.
The issue, I think, probably that's most of interest to people is this one of adverse effects. And I mean, there's different arrays, different ranges of ways in which you can tackle that, because it's a big topic. But we're talking about the mRNA vaccines in particular. I think also the AstraZeneca, the viral vector ones, had its own. But what are we seeing and why? And are we seeing any more awareness in the community, the medical community and the general community, think about some of the problems that have arisen? We've seen, you know, a range of now admitted adverse events coming from some of these therapies. Talk about Pfizer and Moderna in particular. Is the scientific community, the medical community, are they understanding this, or where is it up to?
So I think whenever we're talking about, you know, safety of vaccines, everyone needs to understand that it's really a question of risk benefit. So there is no such thing as, a perfectly safe drug or a perfectly safe anything.
Yeah,
I mean, it's not perfectly safe to walk across a road or walk out the door or even be in your house. Might fall down on you, have an earthquake. So we should never pretend, and unfortunately that's what's happened in the last 3 years, we should never pretend that that's an absolute. There's such a thing as this is absolutely safe. It's not. It's, the question you should be asking is the proven benefit of what we're proposing to do here outweigh the potential risks? And normally that's a decision that would be taken by someone expert in the area for an individual, because every individual has a different benefit and they have a different risk. So it all has to be done at the individual level. That's why we created doctors. That is actually why we have doctors. Otherwise, why don't you go to a chemist and just pick up a prescription for what you think you need? Well, the answer is... I do that,
by the way.
Yeah. Well, but the reason we don't allow that, is because, even if it's a cholesterol-lowering drug that, is considered safe, relatively, it has side effects. Unfortunately, it could kill some people. The benefits, again, are going to be very different. I mean, the benefit of cholesterol lowering in a 60-year-old guy with angina is dramatically higher than would I give it to a 10-year-old perfectly healthy child. If I did, I should be struck off because there the risk, even though it's small, the cholesterol-lowering drug is not going to kill every child. It might, kill 1 in a million. But there is the benefit is zero, and therefore that is completely unjustified for someone to give a cholesterol-lowering drug to a healthy young child. All right, same drug. They're actually culpable if they don't give it to the 60-year-old guy with angina. Same drug, same safety profile, very different benefits. And in one, the benefit massively outweighs the risk. In the other, the risk massively outweighs the benefits. That's the same true of vaccines, but we're not hearing it. We're not being told that. We're not allowed to discuss that. Bodies such as APRA have said we're not going to allow doctors to actually comment on that or discuss the whole concept of risk-benefit, that this vaccine may be a benefit in that individual, but in this individual, young, healthy child, there is no way that you can claim that the benefits outweigh the risks.
And that's the case in, the younger groups, the sort of more 5 to 45 perhaps, or even a different range, but perhaps not so much in the 80-year-old bracket where there might be more of a call. So, I mean, that is really interesting, isn't it? That age group, that youth age group, appear to have had a higher problematic rate with this vaccine. We're seeing, we showed it through an FRI, the presentations, the cardiac wards in South Australia spiked in those age groups around the times of the injections becoming online. The risk benefit in those groups must be minimal. It must be sort of, like a poor.
Well, I think you're trying to say that the risks probably outweigh the risks
outweigh the benefits of the, Would that be a fair statement?
Well, again, that needs to be assessed at the individual level.
Right.
And so we can't generalise, and that's why doctors don't generalise. They ask you a medical history, they assess your risks, and they assess the known benefits, and they make the calculation on every patient they see. They're making a new calculation. They're not going, I don't need to see you. Take this. Do you know what I mean? A doctor will spend a lot of time getting the information that they need to make that. So the same is true of a vaccine. They're a drug. It's a drug. And so, what should be happening is someone needs to be individually making that risk-benefit assessment who has access to the right information, the true information, not, information claiming, well, this is safe and effective. Well, that's not information. That's propaganda.
That's a slogan.
Because it should say, well, the benefit is, this will reduce your risk of, say, death from COVID in a young person from 0.001%. Even if it was a 50% reduction, it would go to 0.0005%. And they go, but so you're saying my risk, even if I don't have the vaccine, is 0.001%? That's not much of a risk, is it? But, in terms of the virus, but so what's my risk of getting a side effect from the vaccine? Oh, well, maybe that's 1%. Oh, so you're telling me now my risk of getting a side effect from the vaccine is 100 or 1,000 times my risk of getting a serious consequence of the infection. Oh, in that case, I think I'll pass. Completely legitimate personal decision. Because they're given the true information, which has not been given. In fact, it's been suppressed. And then they're making a decision. Now, some people who are risk averse say, look, I accept that they are the numbers, but I'm going to give it a go anyway. Just like someone who's told your chance of winning Tatslotto is millions to one, it's pointless buying a ticket. They're given those facts and they go, well, I know that, but I'm going to do it anyway. And so some people might make that decision and have a vaccine, even if the risks overall probably outweigh the benefits. Other people, of course, are going to logically say, well, no, given that information, I'm going to choose for my personal, body. It's called bodily autonomy. We all have a right to determine what goes into our bodies. I'm actually going to pass on this occasion. I might change my mind if the virus becomes more virulent or if a vaccine's brought out that has less side effects. But right here and now, based on the actual facts, the science, I'm going to make a decision about me. That's what medicine every human on this planet should have a right to do, and it's been taken away. That is a dreadful situation.
I totally agree with that. And just in terms of the process then for an approval, this was given provisional approval by the TGA. That's a relatively new phenomenon, the provisional approval process for the TGA. That goes back to 2018?
Yeah, so this was something that was introduced, I guess, for a range of scenarios. One was that particularly cancer patients were getting upset that cancer drugs were showing benefits in clinical trials, but then they were being told, well, the drug won't be approved for another 5 years. Because it has to go through the normal TGA or regulatory process, and that's how long it takes. And they're saying, but I'm going to be dead in 12 months without a treatment, and you're telling me I'm not allowed to have a treatment that's looking very promising because you want to take 5 years to think about it. So they said, oh yeah, look, politically this is really challenging to deal with. Let's bring in a process for life-saving drugs where basically we don't have to wait that 5 years. We can actually approve them just based on some early data that looks promising, and then we'll continue to collect the data after subsequently. And if it turns out not to be true, we can withdraw the approval. And if it turns out the drug is effective and it's safe, in 5 years' time it will get a full approval.
Right.
So that was the scenario in which it was introduced. Yes, it could also be used on a vaccine in a pandemic, but ultimately it was more driven by the idea of someone with cancer needing access to a treatment urgently, having promising treatments available, but them not being allowed to have it because of this long regulatory process.
And so, I mean, we've now seen more information coming out about the adverse effects. We haven't talked about them very much, but they are things like heart inflammation, they're things like thrombosis, blood clots, those sorts of things, strokes, autoimmune problems. Based on what we've seen now, and don't answer this if you don't feel like it, but are we seeing a framework of evidence that suggests that these vaccines should be pulled from approval now, in your view, or the mRNA vaccines in particular? Are we seeing enough evidence now for that provisional approval to be reconsidered?
So again, go back, risk benefit. So people calling for something to be approved or something to be drawn, that's not the way to think about it.
Right.
Because, you may have a drug that, is a lifesaver for one population and lethal in another. Would you say, because it's lethal in this group, pull the whole drug off the market and don't allow these people to use it as a life-saving treatment? That is not the way. The way you look at it is risk-benefit. What are the risk benefits? Are there groups in the population that shouldn't have this drug because for that population or group of the population, the risks are so much greater than the benefits, or that it can't be morally justified. And yet at the same time, you may have benefits far greater than risks in others. So it's not about is a drug or vaccine approved or not approved. Normally, with a drug, if you look at the fine print, you'll find these restrictions are already built in. So it will say this drug is approved for people over the age of 60, or this drug is approved for people with this particular cancer. So that is the normal way it's done. So the regulator is automatically doing the risk-benefit and then segregating out the people or population where it believes that doctors should have a choice, but still the doctor is the gatekeeper. So the regulator is saying, here's a cancer drug, can be given to people with lymphoma. They're not saying this cancer drug should be given to people with lymphoma or that this is a really good thing to give to all people with lymphoma. What they're saying is we think that, there's enough evidence that a doctor should be able to decide, after looking at the patient, after looking at the drug and what its benefits and risks are, he can decide, is it suitable for that person? So we're just giving them the opportunity. The regulator isn't saying, and a regulator is never saying, except I think we have a regulator who crossed this barrier in Australia, this drug is safe and effective. That is not what a regulator says. Based on the evidence the company has given us, there's a reason to believe that it's safe enough and effective enough in potentially in part of the population that an individual doctor should be ultimately in a position where they can make a decision to give this drug or not. So a regulator does not prescribe drugs, and they do not, and I'll say this to John Skerrick or anyone, they do not determine whether a drug is safe and effective, because safe and effective is only relevant to the individual to which the drug is being prescribed. The TGA are not sitting with you, taking your medical history and then determining this drug is safe and effective for you. Only your doctor can do that. The TGA can't. They need to back off and do what they're meant to be doing. And I think they've crossed the line.
In terms of one of the great, push points for mandates in the workplace and elsewhere was transmission, this issue of, you're protecting grandma if you take this, Just starting from a point before that, though, vaccines generally. Now, what's the profile on transmission for regulation vaccines? Starting back to the early adopters, I think there's a conception out there that transmission is nipped in the bud by having a vaccine, but that's not always the case or ever the case. So
transmission is actually the hardest thing to achieve with a vaccine. So the majority of vaccines are designed to stop the infected person getting terribly sick. All right. Now, if a vaccine is incredibly effective, as well as stopping you getting sick, it actually stops you getting infected, which is what we call sterilising immunity. And if you don't get infected, you're obviously not going to transmit it.
Right.
as well as that, maybe if the infection is really mild, you'll still get infected, but there's so little virus because of the vaccine, there's not enough virus to transmit to another person. So the normal hierarchy is, a vaccine, will be expected to reduce the severity of disease. Like if it doesn't do that,
it's not doing anything.
Well, it shouldn't be approved.
Right.
And if it achieves that, that's enough to get it approved. Our vaccine reduced, severe disease by 78%. So, that's a hard number. It's working. If you get downstream benefits like on transmission, they're not, prerequisites. And they're by no means definite. And it was very clear in the very earliest days of COVID, and in fact, we already knew this because of similar mRNA or RNA viruses like influenza. It was highly unlikely that COVID vaccines would ever stop transmission. And we had people like, Professor Ian Fraser, who helped create the HPV vaccine in Queensland. I think he got into trouble because he went to the press, very early in the pandemic and said, look, I don't think COVID vaccines are ever going to work. And certainly they're not going to prevent transmission. And then he went silent. And in fact, it turns out he was absolutely right. And the rest of us thought the same. We're developing these vaccines in the hope they prevent death and serious illness. And that was all we were really hoping for, because we were thinking even if we get that will be a big achievement, because Ian Fraser doesn't even think we'll get to that point. Do you know what I mean? So none of us were expecting an effect on transmission from day one, while we were still designing the vaccines. And then when we saw the first, clinical data coming out, whether it was the adenoviral vector vaccines or the mRNA vaccines, it was blindingly obvious. Why was it blindingly obvious these were never going to stop transmission? Because they're saying, these are reducing severe disease by, 90% in those very early days, because they're up against Wuhan, which was a weak strain. So 90% was good, right, for severe disease. But then you're digging down into the data and you go, but where they've measured protection against infection, it's 50% or less. Well, yes, that's a benefit, but it means 50% of people who are vaccinated are still going to get infected. 50% of people who are vaccinated, even that best case scenario right back there at the very beginning, when the vaccine matched the virus, it was a weak virus, they were picking healthy people to vaccinate, so everything was in their favour. Still, half the people who were getting vaccinated at that time were still getting infected. Didn't mean the vaccine was not doing anything, but it said it was completely useless, because when you want to prevent transmission, you have to prevent all transmission. You only need one person to be protected, and the virus will still transmit through the community. So the nonsense that came out, from some of the institutes in Australia claiming get to 70% coverage and the pandemic's over, there'll be no transmission. I mean, that was ludicrous. A high school student should have been able to logically work out that was a ludicrous assertion that had no scientific basis.
And I mean, this was part of the issue about mandates in workplaces, wasn't it? That, you'll protect your workmates and but you're never going to do that. We were never going to do that.
No.
I mean, things that might have actually helped would be what? Air conditioning, better ventilation, those sorts of things.
Absolutely. In fact, the science, as they call it, because I think I've never seen science so abused in my whole life, and what they claim to be the science, which is about as anti-science as you can get.
Very true. Very true.
And the facts that they've tried to put out that they say are scientific foundation had no scientific foundation. But certainly the science shows that, the things that affect an aerosol transmitted virus are things like, as you say, ventilation, that people were much more likely to get infected if they're indoors in a closed environment that had poor ventilation. The minute they went outside, or if you put in better ventilation, their risks of transmission went down. I mean, again, not rocket science, But, did they invest in improving air conditioning and airflows and looking at that? No. We're going to go this other way. It sounds easier. And
the concept of the pandemic of the unvaccinated then was always a nonsense.
Well, it wasn't just a nonsense. I mean, it was I think, a disgraceful campaign of victimisation and personalisation, which had no scientific basis whatsoever. It's a form of racism. It's just labelling people for the sake of labelling them for political purposes. And let's be honest, it was all about the political purpose. It wasn't about stopping people dying or stopping transmission, because we knew that that assertion was completely false scientifically. Do you know what I mean? So it was just about, I can get political advantage by labelling a group of people, making them the enemy. Do you know what I mean? Well, again, in history, when that's been done, it's again done for political purposes, and it has enormous implications and ramifications for those people you're labelling. They're going to suffer. And that is, how that was allowed to happen, I don't know. It was disgraceful. It shouldn't be allowed. And I'm disappointed more of my medical colleagues didn't speak out against it. In fact, a lot of them were the ones who were propagating this false message that somehow unvaccinated people were a threat to others. That was never the case. The science doesn't support it to this day. And what they were doing was totally despicable. And as I say, those people should be called out and held to account, because that is not the way that you run public health.
The whole thing has become, there's just so many limbs to it now, and we're sort of getting, and I don't want to hold you up too much longer, but I've just got a couple of other questions that I'm really keen on. There's been a recent study, I think, I just don't know exactly the name of the journal, but I read an article about it recently about we're now seeing an IG4, a problem with the suggestion, in fact, and perhaps you can give that a level of scientific credibility that I've just killed. The IG4, this immunoreaction, that the suggestion that the vaccine actually may be causing an immunity problem for those that have had 1, 2, 3, 4 doses. Is that real? Is that a real thing? Is it a problem? And there's also this sub-question then in there about this concept of leaky vaccines, which I hear occasionally, and what it's doing to creating new variants.
So the situation of IgG4 being induced by mRNA vaccines specifically, is a fascinating one. It's very real. So the science it's published, it's peer reviewed.
Peer reviewed, okay.
By very credible groups. So it's happening. Was it expected to happen? No. What does that tell you? We don't know anything about this new technology. The claim that this is an established, understood technology, that in itself proves the lie. Do you know what I mean? This is an experiment. It's an experimental technology. We still really don't know much about it and what it might do. That's scary. And that's why all of us were terrified when this was rolled out. No joke. As vaccinologists, we're all discussing this in meetings and getting together, and we're all going, this is super scary. We don't think the people who are driving this, the politicians and some of these public health people, have a clue what they're doing. They're rolling out a completely experimental treatment where we have no understanding of what it's going to do or what it might do. And they're proposing to not just give it to a limited number of people, they're proposing to give it to everyone on the planet. And then work out what does it do and what might it do. And you're going, this has never happened in human history. It's the most insane experiment, is you're putting the whole population of the world potentially at risk. And then you go, I hope it goes well. I hope in 10 years not everyone on the planet keels over. Hopefully that won't happen, but there's no guarantees because this has never been done before. This is a completely new technology. So when I see something like, in the recent months, mRNA vaccines are inducing this antibody called IgG4. No other vaccines induce IgG4. What's IgG4? So I know IgG4 because I work in the allergy field. So IgG4 is an unusual antibody because it's not designed to protect you against viruses. Quite the opposite. It's an antibody that's designed to switch off the immune system if you have a protein in your body that doesn't go away. Because viruses, if the immune system sees a virus and attacks it, after a while it should go away. If whatever the immune system attacking doesn't go away, either the immune system keeps attacking forever, and that will eventually kill you, your own immune system. So the immune system has a mechanism. Well, if something stays around too long after I've attacked it, I decide I'm going to ignore it because that's safer, even if it turns out to be some chronic virus. And we do have chronic viruses. The reason we don't get killed by our immune system is if something stays around too long, it switches off. And IgG4 is part of that process. So in the allergy field, when we're trying to switch off your allergy to, say, bee stings, we're actually trying to make your body switch over from IgE, which causes the allergy, to IgG4. Which basically just switches everything off and you stop having allergic symptoms. So it's not something you'd be trying to design any vaccine to induce, maybe an allergy vaccine, but not an infectious disease vaccine, right? The answer is no one knew this was going to happen because they'd never studied mRNA in reality in humans before in any detail.
Would it have been at all possible to know that as a possibility? I mean, with that set of sequences you've talked about there, is there any way you could have known?
Not unless you'd studied mRNA before, which no one had. They claimed they had, but they clearly didn't. Otherwise, they would have told us this is what's going to happen, and they didn't.
Yeah.
A bit like the myocarditis. If you understood mRNA, they would have told us, mRNA, there's probably going to be a small number of cases of myocarditis because we've studied it before and we know this happens. Did they tell us that? No, because they had no idea because they'd never studied it. The claim that this technology was well studied and understood is a complete and utter lie, right? It had been around, it had been tested in a few mice, and had largely been discarded. Most people have decided it's not, as I say, suitable and it's not ready and we don't know enough about it. So to now reinvent the whole story and say, oh no, this was a really well-studied technology that had been tested and we knew it was safe and effective. Well, these are the facts that are proving the lie because the fact myocarditis wasn't predicted meant you weren't telling the truth when you told us this was a really well-studied and well-understood technology. The fact that we're seeing all this switching in humans to IgG4, which is completely left field, means you were lying when you told us that this was really well-studied and well-understood. It clearly wasn't. So these are just signals that are telling us we still don't really understand this technology. Now, when you say, is that a good or bad thing? The answer is we don't know. Like so many other things with this technology, I can't give you the answer. Now, I can speculate and say, well, to me, it doesn't look like a good thing because IgG4 is not a great antiviral antibody. We need IgG3 for that. Now, our vaccine, fortunately, makes lots of IgG3, which is the antibody you want if you want to fight a virus. What this IgG4 is doing, we don't know. But to me, it's a really bad signal. One, we don't understand the technology. And it may mean, as you say, unfortunately, the mRNA vaccines are going to give you less and less protection as time goes by. And in fact, the evidence is that that's what we're seeing, that if you have 3, 4, 5, 6 doses of mRNA, it seems that the amount of protection you get is more and more transient, if anything. And so you are getting into a law of diminishing returns.
Is it damaging immune systems in relation to other infections, though, or or is that still unknown? For example, if you have your 5th booster, which is the target you're now toying with, I see, which it's a comment for a different day, but is that lining people up for, potentially for a weaker immune reaction to the common cold or something else or, whatever it might be? Or is that something that's just related?
How can we possibly know?
We don't know.
It's never been studied.
Yeah.
So we're told that these were studied comprehensively. Right. But unfortunately, that was a lie. And so we're left with a situation where I can't answer your question. There's no one on the planet knows what the consequences are now going to be. We're just going to have to wait and watch and hopefully collect the data and see what pans out. I mean, it's really scary because the answer is we simply don't know.
Are we likely to see other left field, IgG4 type consequence sequelae that we haven't seen yet, that we don't know about yet? Is it possible there'll be, other things that are emerge?
I think almost inevitably there will, because, again, this is a completely different technology to what we've ever used before. Like all new technologies, you never cease to be surprised at what you didn't know or didn't expect. And it doesn't apply to medicines. It applies to any new technology, like even computers throw up surprises. So again, I think, we're in this area of the unknown where we shouldn't be presuming anything other than there will be other surprises with mRNA.
It is genuinely fascinating stuff, and I'm conscious of time. I want to ask you this last final question about, we're seeing now fluctuating around the line, but between 16 and 17% excess deaths in Australia. That's a pattern that's been largely repeated in countries that adopted the mRNA technology. Is it it is a very unusual time. We all accept in the last 2 years, lockdowns, people in their houses, people using alcohol and all of your other things, delayed lists in hospitals and all those sorts of things. But the media don't seem to want to join the dots and suggest that somehow there might be a correlation between mRNA vaccines and that excess death rate. Do you have a view on that, or is that unscientific to draw that bow?
Yeah, well, I think that is a bit of a long bow. I think what's clear is the excess mortality rates are real. Those signals appeared about a year ago. They've now appeared in most of the Western countries. They are way off scale in terms of statistical probability because, mortality does fluctuate, but they're way off scale. So, they're real. More people are dying than ever before. What's driving that and why it's happened in the last 18 months, at this point, you can only speculate about. So we can't say definitively what it is, but it is really happening, and it is very, very concerning. But I guess the problem, like all these, is if you don't allow a particular question to be asked. Right. So you say, I'm going to let you ask questions of, is this because of lockdowns, or is this because, people didn't get appropriate treatment or had too much alcohol? But I'm not going to let you ask that other question I can see you want to ask. I'm not going to allow that to even be scientifically questioned or data collected on it. That's what I call censorship and scientific censorship, and it has no role in science. You cannot shut down one question because you don't like it and not explore that as a hypothesis. So at the moment, your question, I would say, well, it's a reasonable hypothesis. Certainly, yes, the vaccines only appeared in the last 18 months. If excess mortality rates have only increased in the last 18 months, it's a reasonable hypothesis that someone now needs to go out, try and collect data and determine is there support for that hypothesis or not. But no one can say you're not allowed to have that hypothesis. That's again saying to a scientist, we'll let you have this hypothesis. We're not going to let you test that one. I mean, that is not science. That's not legitimate.
And what does that look like in terms of the actual data collection? Is that an easy thing to study from a data point of view, or is that is it a difficult thing to do now? Do you need autopsies? Do you need, or is there other mechanisms? For those that might be listening to this in the political sphere, what are the sort of things that should be done that are not being done from the public health perspective to study that, or is that something for the private sector?
No, definitely not the private sector. So proving causality, so showing one event caused another event is incredibly difficult. So the answer is you can only do it by collecting every piece of conceivable data. And I guess I would take you back to the example of cigarettes and lung cancer. So that went round and round, for decades with obviously large numbers of scientists saying, no, there isn't a causal link between smoking and lung cancer. And obviously the cigarette lobby were heavily involved behind the scenes in buying those scientists. And you could argue maybe that's now happening, with the drug industry. Because again, they have vested interests. But it took 20 to 30 years before finally, from every conceivable source of data, including, making monkeys become addicted to cigarettes and smoke their whole lives, and showing, they were getting increased risks of
on roller skates with a fez that's smoking cigarettes.
But those experiments were done because no one would believe it, and of course causing cancer takes 20 years.
Yeah.
And so, you have to run those monkey experiments for 20 years before you have the data. Oh, actually, the monkeys are all getting lung cancer now. But, so the answer is it's incredibly difficult. You have to collect every form of data that's conceivable and put it all together. And that's not being allowed right now. Which is the concern. So as you say, yes, everyone who's dying after, having a vaccine should have an autopsy. That should be the default. They're refusing to do autopsies. Why are they scared to do autopsies?
Is that delaying? Is that upsetting the apple cart in terms of ever being able to find out, specifically what that person died from? I mean, is it, you would never know. If you don't do the autopsy, you never know. That's why in the old days, autopsies were done on everyone pretty well who died, certainly in a hospital, who died or as a young person or died in unexplained circumstances, unless there was really a obvious cause. Reason, this person has cancer, and finally they've died. Autopsies were routinely done. Nowadays, it's only if you suspect there's been a murder case or something, it's gone to the other extreme. But I think here we're in a situation where we desperately need more data, and we should be doing more autopsies, no question, and particularly in these people where otherwise we can't explain what's happening. And I think, there was just an autopsy series released from Japan, from Tokyo, just in the last week, where they are now routinely trying to autopsy people who die within a week of vaccination, along with other people. And they presented a case series of 50 people, of which a number turn out to have severe myocarditis. They're young males with no other disease cause. They were clearly killed by having an mRNA vaccine. I don't think, And then it's not a fact that this isn't happening. What we need to know is how frequent is it? How far?
Is it 1 in a million where we might say, okay, that's an acceptable risk? It sounds bad, but it's acceptable because maybe we're preventing, again, 10 deaths from COVID. But if it turns out that risk is 1 in 100,000 or even, 1 in 50,000, we know that you don't have that risk of dying of COVID. So that becomes really, really concerning. We need that data. We desperately need it. And we're only going to get it by autopsying these people and determining what is the true frequency of this side effect. Let's stop pretending it doesn't happen. Which unfortunately till recently, again, regulatory bodies, TGA, were telling people there have been no deaths, in Australia caused by myocarditis, right? That was never true. And now it's been established, and they're changing the narrative, and they've now, accepted one case. That's the tip of the iceberg. We need to know what's the true frequency that this is happening, because we can't work out, how do we inform someone of the risk and benefit? If we haven't quantitated the risk. We'll just pretend it's infinitesimally small. We don't have evidence to support that. In fact, the evidence probably goes the other way, but we're going to tell you that anyway. We're just going to lie to you because it's in your best interests. So, public health now officially has the right to lie. I don't think public health should ever have the right to lie. Just lie to people to get people to do what they want them to do. That is not a democracy. That's not a civil society. That is an authoritarian totalitarianism that I don't think has any place in Australia, if not globally.
I agree with you. Now, I want to ask you one final question. Is there anything that's still around that we haven't blamed long COVID on yet? Is there something that long COVID hasn't caused? Change in weather, is there something you can pin it on? Because long COVID seems to be getting blamed for everything. And is it a thing?
Well, I'm trying to find someone who has long COVID. I know thousands of people who've had COVID. I haven't yet found one individual who tells me that they have long COVID. So, when I'm told 20% of people who have COVID get long COVID, I mean, it's complete utter rubbish. I mean, you just survey the people around you, you know it's rubbish because you know everyone's had COVID. If you don't know anyone with long COVID, well, then that tells you the frequency must be less than the people you know. So if you know 100 people, no one's had long COVID, it means it's less than 1%. I suspect it again is even much lower again than that. So yes, it's being used because unscientifically the frequency is being grossly exaggerated, just like we saw with case fatality rates, because they're not using proper denominators. And they're obviously over-diagnosing it, and they're claiming anyone who has a headache, a month after COVID has long COVID. Well, most of those people were probably having headaches before COVID. A lot of long COVID is again, people who have particularly a flu or a serious viral illness. It's normal sometimes to take 2 to 3 months to recover. We don't talk about long flu. You can't work because you've got long flu. You know what I mean? And yet we know flu actually, medically we know it does that to a lot of people, that they're incapacitated for much longer than just the week they have the infection. But again, so it's being used for political purposes. I'm not saying that, some people don't have true prolonged sort of disability from having an infection, just like with influenza. That's not what I'm saying at all, but I'm saying the whole message around long COVID's being exaggerated for political purposes. But I don't think it's founded in the science or what I've observed, from the medical side, that we're not seeing people, 2-3 years after having COVID who incapacitated. If they exist, then they're very, very small numbers and can't be used to justify taking everyone's rights away from them, which is what's happening. Because these are all excuses at the political level to take away people's rights, let's be honest. Force people to do what politicians want them to do for whatever reason, because they're not allowed to question those reasons. That again is not democracy. As I know it, and that's why we have to push back and say, no, the science should be dictating policy. Here we have policy dictating science. That is never going to end well.
Well, hear to that. And look, thank you for coming in and thank you for sharing all of that. I hope it's very interesting to me. I hope people will find it interesting. I'm sure they will out there, and it's informative as well. I know people are looking for these answers. And so, look, thank you for taking the time to come in and talk to us and be the first victim of the new podcast. You've done well. You've survived. I think we've survived. You've survived. It's hopefully we got it all. Otherwise, we've just been sitting here talking to each other, which has been good as well. So thanks again and all the best. And we'll be watching with interest to see where your vaccine goes and for your exploits in the future. So thanks very much.
It's been a pleasure.
Yeah, appreciate it.